And oh, this is being recorded. So the picture that you see there is from the city of Arequipa. This is downtown of Arequipa, Peru. This is the second largest city in Arequipa, and this is where my research is based. But I'll start with this simple question that I was asked all the time when I was a little kid in Lima. I'm from Lima, from the capital of Peru. And I remember that I always said, I want to raise animals. That's what I wanted to do. I didn't know if that was a profession or anything like that. And I remember, and I found a book, one of those books that you have in high school with friends of yours writing about you, you know, you're developing your personalities and all that. And a friend of mine, he said, you are going to be a veterinarian. That's the only thing that you care about, you know, animals. So you're going to be a veterinarian. But the thing is that, you know, the path to get where you're going to be is never straightforward. And especially in global health, I think that there is never like a straight path to global health. People do other things and then they start working in global health. And that was the case for me. I finished high school. I moved to another city, a small city in the north coast of Peru. And I didn't know what I wanted to do. So I joined this school of, it's a career that it doesn't even have a translation, but it's Ingeniería Zootecnica, which translates pretty much to engineer to produce meat. So yeah, I was going to learn how to raise animals, but not for the reasons that I wanted. So I spent just 1 year in that school. I spent 1 year and then I was spending too much time playing soccer instead of going to classes. So I realized that I had to do something. So I moved back to the capital, to Lima. And then I thought, maybe I want to save animals. I want to save wildlife. And for that, I need to study conservation. And I found this school, which is a school of forestry engineering with a specialization in conservation. And I joined that school. So that advertising was completely misleading because 90% of what they do is to cut trees to produce wood. But I spent 3 years in that school. It wasn't wasted time because I learned calculus at the engineering level. So I left that school and then I realized I was much older by then. It was already 4 years of college and I didn't finish either program. So I decided to be a veterinarian. I applied to a vet school. That was my third college, right, at that time. So my father told me, okay, you're on your own. You know, I've been paying 4 years of college education and now you're on your own. So I started selling dog food. I, you know, put a nice shirt, you know, nice khakis and got my briefcase and I started visiting veterinary clinics and working with a guy who was importing dog food from Spain. And I was just doing that for like 6 months. And I saved enough money to get into the institute, because you need to go to an institute to prepare for a few months before you go to vet school. So I did that. I entered, and I got accepted first place in the vet school, in this vet school in San Marcos University. This is called the Dean of American Universities, American in the sense that it's from all the Americas, because it's the oldest university in the Americas. It was founded in 1551. So I joined the vet school there. I was extremely excited. I was still not receiving any money from my father. So my mom was putting together all my exams with all my very good scores, you know, for the first semester. And then, you know, she gave him this package with all the exams and these amazing scores. And he started giving me money. That was good because I was already running out of savings. And then within vet school is when I realized that I wanted not to save just animals. I wanted to understand more complex issues such as zoonotic diseases in public health. I started working with is the cysticercosis working group in Peru. It's a huge group now. They started being funded by Bill and Melinda Gates like 25 years ago, and they built a huge operation in the North Coast where cysticercosis, this disease that is transmitted between pigs and humans, is a parasite. It's not a, sorry, the disease is not transmitted. The parasite is transmitted between pigs and humans. You know, it's very, very prevalent. In some, this is the tapeworm that when the larval cyst goes to your brain, causes seizures. And there are some districts, some communities that we visited, and 5% of people have epilepsy due to these worms growing in their brains. So it's a very sad picture when you go there and you see this, you know, little kids, you know, playing with garbage, you know, making fires, and the pigs are there, and it's and there are no bathrooms, right, or latrines, so people defecate outside, you know, openly, and then the pigs, they eat the feces with the eggs from the tapeworm, and the cycle completes. So I was lucky that in my third year of vet school, I took this very good course on veterinary epidemiology with an epidemiologist that was really well trained. He's Peruvian, and he's a big thinker, and he invited me to join his team, at least for the summers, and to go to this place. He said, we need always volunteers, and I was just volunteering. Vet school in Peru is 6 years. It's 5 years of classes and 1 year of clinical rotations. So I started at the end of the third year, and then I didn't stop. I was all the time in his lab. Every time that there was an opportunity to go to the field, I was just immediately packing and going there and helping. Sometimes we were interviewing people. Sometimes I was taking blood samples from animals. And for me, the most fascinating thing was to start mapping things. I started mapping things, and I did my DVM thesis on the spatial relationship between the human carrier of the tapeworm and the animals carrying the larval cysts, the larval forms of the tapeworm. And I was just fascinated with this idea of going to the field, seeing what's happening, and then being able to put everything in a map and being able to see that. So I decided I'm not going to study wildlife conservation. I'm not going to put, you know, a pet clinic, you know, and be the usual vet, you know, who is always just receiving pets. I wanted to do something different. And then I found veterinary public health. That's a branch of public health, or a branch of veterinary medicine. And I wanted to be a public health veterinarian. And so when I finished my thesis, I received my DVM degree, and I stayed with the same group. And within the same group, there was a smaller team working on echinococcosis, another fascinating parasite that uses this interaction between humans and animals, in this case, sheep, to complete their life cycle. And they also use dogs, actually. They need, actually, the cycle is between dogs and sheep, but we humans, sometimes we accidentally get infected with the tapeworm, and this causes an awful disease, because the larval cyst now can grow in our lungs or in our livers, you know, and sometimes in some areas you can find a lot of little kids with these worms growing in their organs, and they need surgery. The treatment is not great, so usually surgery what they need. So I started working on this and understanding better how pathogens use this human-animal interaction to survive. That's what they're trying to do, right? They're living beings, and they're trying to survive, and they use these interactions. And so I was there. I was working still in Lima. I was living in Lima, working with this team. And then at some point, I wanted to do something different. I was a few years working with them, but I was already volunteering for many years. So I wanted to do something different. And I was in the most bohemian district in Lima, called Barranco. and I was in my friend's birthday party. And then she introduced me to this guy, American, very nice guy. And we started having a couple of beers talking. And he told me, I have a project in the south of Peru about Chagas disease. And I'm like, I'm looking for a job. You know, I'm a veterinarian. I'm interested in zoonotic diseases. And he's like, I want somebody who knows about maps or is interested in maps. So he hired me. And long story short, this guy is Michael Levy. Michael Levy, he's a professor of epidemiology, an associate professor of epidemiology, also at the Perelman School of Medicine. And that was in 2008 that I met him. Well, we had a formal interview, not at the bar, but a few days after. And then I moved to Arequipa, to his study site. And I moved to this place with my wife. Believe it or not, this is a huge coincidence, but she was already discussing with her boss about moving to the same city. And turns out that Mike and her boss, my wife's boss, they were co-PIs in the same project. It was a huge project, and this other person was in charge of the qualitative and more the human side of the project, and Mike was more in charge of the animal and vector side of the project. So when I told my wife, we're like, okay, we have to discuss this. We have these opportunities, and then we realized it was the same project. Huge coincidence, and we immediately packed and moved to Arequipa. And in this area, I was studying this different system. We call these systems, right? When we talk about in disease ecology, this is a system. We're studying the vector, we're studying people raising guinea pigs in this area, and these are the animal reservoirs. Guinea pigs, you know, they're very prevalent, they're very common in Peru, Ecuador, and Bolivia, because they are raised not as pets, but as sources of protein. So you can find these farms, you know, like the one you're seeing there. And sometimes these vectors use these animals as sources of blood meals. And then you have the parasite in the vector, so the whole cycle can be completed. Now, that site in the city of Arequipa is actually, it's a consortium. It's a site that is shared between a Peruvian university, Cayetano University, and Penn. And here we conduct multiple projects on infectious diseases. She is Sherry Sie. She is a DVM student. She's finishing in a few months, and then she's going to join us to do a postdoc. She also finished last year her PhD, her PhD in epidemiology at Penn. And in that picture, she's in Arequipa also, and we were putting GPS collars on these alpacas to understand how they move and how that might influence the transmission of this parasite called the liver fluke. So that was a lot of fun. Of course, that was pre-pandemic. That was like 4 years ago, or maybe more. But in that area, we're studying the liver fluke, Chagas disease. Now we're also working on hydathic disease, because that is also prevalent in Arequipa, in that area, and rabies. And I will talk more about rabies because that's my passion, and that's what I'm spending most of my time now. But you can, I hope you have heard about the One Health approach before, but if not, the One Health approach is understanding that environmental health, human health, and animal health are always connected. And if we study them as a whole, we can find a lot of synergies. We can collaborate with our people. We can we can reduce effort. You know, we can avoid duplicating efforts, and try to create a healthier planet. And this is a very fitting lens, you know, to study zoonotic diseases, and we will see how later. But this is the approach that I use every time that somebody talks to me about a disease. Now, I'm not thinking just about the disease and compartmentalizing the disease, but I'm trying to understand all the different connections that the disease of the pathogen has with humans, animals, with the environment, or with any other aspect that might influence the disease. So as I mentioned, Chagas disease, it's a vector-borne zoonotic disease, which means that it's complex, right? Because you have all these elements playing together. There are insects, there are animals, and of course there are people. And to add to this complexity, this is not like mosquito-borne diseases. Mosquito-borne diseases, the mosquito gets infected through the mouth, and they infect other people again with their saliva when they bite. These insects, awful insects, are called kissing bugs because they usually at night when you are sleeping, they're like the size of a small cockroach. They crawl onto you. They look, you know, they follow usually CO2. So when you're breathing, you know, that's the cue for them to find hosts. And that's why they are called kissing bugs, because sometimes at night when you see somebody sleeping, they're very close to the mouth. And then they bite. And if they're infected, that blood meal is going to push the feces in the midgut, and they're going to defecate very close to the bite. And the parasite actually is in the feces. So this is called stercorarian transmission. And then the parasite, people scratch, and the parasite can get very close to the hole, and then the parasite enters the bloodstream. So very inefficient. If you think of this, what is the likelihood, right, that those feces are going to be in contact with that specific bite hole? Very, very inefficient. So why is that we still have millions of people infected? Why is that we have millions of animals infected? So that is a question, and that is a recurrent question for me. Why does transmission persist, right? When you're working with infectious diseases, that's, I think, the main question. So they're controlling the disease in Latin America with indoor residual spraying. And in many places, this is what they do, but you can see that it's not that easy because the structures, the construction material is very poor. So you have to spend more insecticide because this porous material absorbs a lot. So it's expensive, it's difficult. So this is a question, right? If we have that that control intervention that works, why is that we cannot control the disease? And to add to the complexity, there are multiple insect vectors, multiple species, more than 100. In Peru, we have dozens of them. Here in the US, in the south border, we also have triatomins. Now Chagas is becoming more prevalent in the south border. In Texas, there are people studying this disease also there. And some of these insects have preference for mammals, some of them have preference for birds, and some of them have preference for even reptiles. So you have to understand these kind of ecological interactions to control well the vector and the disease. But only mammals can carry the parasite, not birds, not reptiles. So that is another interesting characteristic of the disease. And there's some animals, domestic animals, that are well-documented reservoirs, such as dogs and guinea pigs. Humans are also good reservoirs because we live for multiple decades. And the problem with this parasite is that once you contract the parasite, once you're infected, you cannot clear infection. The parasite is going to be with you for decades. So people can continue transmitting to the vector, to the insects. Well, I mentioned that guinea pigs are important reservoirs. I'm going to skip this one. They're important reservoirs because, yes, they're not only raised in farms, like the one that I showed you before, but this is in a backyard. And you can see there are 3 of them in that corner. People just put, you know, some stones and mud, and that is a corral. And because these are really, really bad climbers, they're not going to escape there. So people just give them some food scraps from the kitchen, and they have guinea pigs for a meal, you know, when they need them. But something interesting is that they're not rural animals. You can find them in the middle of the city. People have those small corrals in their roofs, on the roofs, and they raise guinea pigs also there. So now you start seeing urban Chagas, you know, that we didn't have before. Now urban Chagas is a thing. Before it was a rural disease, only a rural disease. And then we have the urban-rural interface, the peri-urban communities, which are very unique. And I'm always, I get always upset when people just develop strategies for urban communities and for rural communities, and they completely forget about the peri-urban communities, which cannot fit within the other categories. This, for example, is a peri-urban community. This is very, very close to the border of the city. They are called pueblos jóvenes in Peru. In Brazil, they are called favelas, and they have multiple names across Latin America. But you can immediately see that the density of people is different, the landscape is different. Of course, the socioeconomic status is going to be completely different. So when I started seeing this and studying, not only seeing, because I saw this when I was studying teniasis and cystosarcosis in the north coast, but now I was obtaining data about socioeconomic status. I was mapping these areas, talking with these communities about their daily experiences, and I started getting very, very interested in social determinants of health. I'm not an expert on that topic, so what I did, I started meeting with people who are experts on that topic, and at some point, I got a couple of them working with me, and it's amazing to talk with them. One of them is a social scientist, the other is an economist, and they give so much context to the problem. As a veterinary epidemiologist, I was trained to think about interventions, to think about intervention and control, to think about study designs. But these are different dimension of health that definitely has to be included in a global health program. And then I was doing experimental infections. I was in the field, but I also like to work in the lab, and I was doing this when you infect guinea pigs, and then you see how much these guinea pigs transmit to vector to insects, and that is infectiousness. I was trying to measure the infectiousness. We repeated this in the lab multiple times, and we found heterogeneity. And then we found that there are super spreaders in the guinea pigs. I put this because it's so relevant to what we are seeing now. And for humans, and especially now in the middle of the pandemic, we're talking more about super spreading events, right? A concert or this church in which somebody in the chorus was infected and by singing very loudly, was spraying a lot, and that created a spreading event. But there are also biological characteristics that allow sometimes an individual to be a super spreader, in this case, to maintain parasitemia for a very long time. So every time that a vector feeds on these guinea pigs, the vector gets infected, more vectors get infected. Another aspect that I needed to understand is how people raise their animals. This is very important because in the farms, that wasn't a problem. The problem is when I went to some of these communities, and I was always visiting the same houses, trying to understand how they raise their guinea pigs. And one day I didn't see any guinea pigs. And I'm like, Ceñito, ¿dónde están sus guinea? I was like, hey, ma'am, where are your guinea pigs? I was always being extremely polite because they're always busy and they don't like that. Sometimes I was taking blood samples from their animals. And they say, I don't have many guinea pigs now, or I don't have any now because there is no rain. So alfalfa is expensive. And then I found this beautiful book. It's a book from an anthropologist discussing and explaining how these guinea pigs are raised with this expansion and contraction of guinea pig populations depending on the rain cycle. And this is fascinating because this tells us that there are population bottlenecks, and this can inform why is that the parasite persists. If we can think that there are super spreaders and there are population bottlenecks, maybe we have these super spreading events in which you reduce so much the guinea pig population, that all the vectors have to feed on them, on those few individuals. And if you have some of those super spreaders, then the parasite, the transmission of the parasite explodes. So that's what I was doing for about 10 years, studying Chagas. No, it was seven years. And then I was, and now I'm going to talk about opportunity, because I was studying this because I was always talking with people and I was interested in these diseases, and they invited me. In 2014, I was doing my postdoc in Arequipa. I was doing my postdoc studying Chagas, and we were supposed to start the largest trial that we had done at that time for Chagas, a huge community trial trying to understand why people don't spray their houses, because some people were not participating in these spraying campaigns, and then the bugs, the vector colonies were growing. So that was scary, and we needed to do something. So we were going to start this huge trial, and then it seemed that we were receiving the 7 plagues at once. First, it was strikes. All the people in the Ministry of Health, they were on strike, so we couldn't start the trial. Okay, fine, we delayed. Then we were about to start, and then these unexpected huge rains came. So, okay, we have to stop because we cannot spray during the rainy season because you lose all the insecticide. Okay, so we stopped. And then when we were going to start, actually we started, and after the second day, we received a call from the Ministry of Health, and they said, please remove all your people from the field because in the same locality, we detected a rabid dog. So I was there, and they said that, and I'm a veterinarian, so I was scared but excited. I was excited. I'm like, okay, there is rabies now here, because we didn't have rabies. That was the reintroduction of rabies. And so that was an opportunity for me, and I started studying that. And in 2015, this was at the beginning of 2015, yes. The Ministry of Health reported the reintroduction of canine rabies in Arequipa, Peru. And that was important in the Americas, in the whole Americas, because dog rabies is well under control. And the program is advancing very well since 1982. There are so many areas that are free of dog rabies now. And this was the first area free of rabies that now had the reintroduction of the virus. So I started studying that, and this is just a map showing the water channels in the city. All this is densely populated by houses, but this is just showing the water system. It's the river in the middle, and these water channels that bring rainwater to the river. And then in 2015, in March 17th, the first case was detected. That is what the triangle represents. And then a few days after, a second case. We already, remember, we moved all our people outside of these areas. And then a second case was detected, then a third case. So then it was clear then by the end of the month that this was not a small thing. And this was not just one case that was maybe introduced from Bolivia or from Puno, which are neighboring areas that they have rabies. There was active transmission within the city. 1 year after, sorry, by the end of the year of 2015, cases were all over the area. And maybe you notice that there is a pattern here, a spatial pattern, but the cases are very close to the water channels. And that's why I like to go to the field. I think that you just receive data and sometimes you miss so much. But I saw this data, there was something, but I was also always in the field. And the people were saying, oh, by 2016, you can see that cases are now on the other side of the river. That river was working as a permeable barrier. Now we know that because of genomics. We're doing genomics to see the virus and the clusters that this virus forms. And we saw that actually this river is working as a barrier because dogs have to cross the river, right? And so they only have three bridges to do that. These are dogs moving along the water channels. They are actually using the water channels as highways. we put GPS collars on more than 20 of these animals, and we saw that they're moving slowly within the street. As soon as they enter the water channels, they just move much, much faster. They move longer distances because there are no cars, there are no people, there's nothing. It's just dog's land. But how do you control rabies, right? Because for me, that was fascinating. I was like, oh yeah, let's track dog with GPS collars and all that. But let's go back to public health, right? We need to control dog rabies to protect humans. This is maybe the best example of One Health that I can give you. Dog-mediated human rabies around the world is not controlled at the human level. It's controlled by vaccinating dogs. You vaccinate dogs and you protect human populations. So that is done by mass vaccination campaigns. By the way, these were also conducted in the US before, but now everybody vaccinates their dogs, or most people here vaccinate their dogs. The dogs are on the leash, so there are fewer issues, and there's no dog rabies here. But in Latin America, Africa, and Asia, ministries of health, they conduct mass vaccination campaigns, meaning that they have to put vaccination sites in some places, invite the community, and people have to bring their dogs there. But coverage is not great. Of course, that's why we have rabies here, right? And vaccination rates are very heterogeneous. So sometimes you have these pockets of unvaccinated dogs, and sometimes that is enough to transmit, to allow the virus to transmit slowly there, because the rabies virus transmits at very low rates. Something interesting is that these vaccination tent locations, the location for the vaccination campaigns, are chosen by convenience or by just what people remember that worked well 20 years ago, and they want to continue doing the same. But populations change, cities are dynamic, so we have to adapt to that. I love this picture because it's clear that people try this. I remember that we were talking with this person. She was bringing her dog to the vaccination campaign, and some people say, well, I don't have a dog leash, so it's difficult for me to handle my dog and to bring it to the campaign. But you can see that she was trying with a plastic bag, she created a harness and she brought her dog. And of course, that's not going to last, and she doesn't walk the dog with that plastic bag all the time, but she did that just for the campaign. But in all cases, they just have to use brute force. And these are very, very different conditions than the one we have in the US. They have dogs that are guard dogs. They're not pet dogs. Many times these animals, they don't have a name. They are outside the house all the time, just guarding the house. So when they have to bring them to the vaccination tent, to the vaccination point, this is what they have to do. And they have to do the groceries. I saw her, she finished with that, she went to the corner store, she bought some groceries, and then she had to go back home pulling the animal. So there are barriers. There are many, many barriers for vaccination in these areas. So my research now is how do we optimize the location of these tents to increase vaccination coverage? And I've been working on this pre-pandemic. Now, it's in terms of funding, there is a lot of interest in my research now because we have seen what has happened during the mass vaccination campaigns here, the mass vaccination sites for COVID. They didn't work as we expected. In many places, they were closed just because they didn't work at all. So now we're going to start a new trial, a large trial, to understand what are the factors, what is what we need to understand about mass vaccination campaigns in dogs to implement better mass vaccination campaigns for humans. And this is spatial optimization that it's what I do ideally can also increase coverage or uptake for other public health services. So now you see, again, going back to this One Health approach, how is that the urban environment influences diseases, the urban environment influences how animals move. Then we have urban behavior, of course, that also overlaps and interacts with the urban environment. And then we have the animal populations, right? The animal populations interacting with the environment, interacting with humans. And in the middle of that are these emergent zoonoses. Now I'm talking about rabies, because rabies right now is an emergent zoonosis or re-emergent zoonosis in Arequipa, but it's emerging in other places. That's what I'm trying to understand. Why is that it is emerging in southern Brazil? Why it is emerging in places in central Argentina or in Chaco, Paraguay? Is a disease that was well under control, but now is being a victim of its own success. People are not vaccinating their animals. Authorities are not paying attention. Policymakers are more interested in funding other programs that they think that are more urgent. All that is important. And within each of these components, you can see that there are dynamic processes. There are dynamic processes such as urbanization, migration, how people raise their animals, if people participate or not in health programs, and then the interaction between animal populations, of course. So all this is fascinating to me. It's really fascinating from an intellectual perspective, but what's my goal now? And I think that in global health, if you are doing global health, it's very difficult to just do global health for a specific research question that is just intellectual, right? Especially if you have been in the field, you know, and you have met people in the communities that you are studying. So my goal is to eliminate rabies. That's my goal. I want to eliminate rabies from Arequipa. I want to expand that and eliminate rabies from Bolivia. I want to also help my collaborators and my friends in Guatemala to eliminate rabies there. And there is a goal now to eliminate dog-mediated human rabies all over the world by 2030. That means we have only 8 years. 8 years. So I showed all this to you to also explain that at the beginning, there are some local questions and interesting questions. And I was following them like, how do animals move in the city and things like that. But is that going to help me eliminate rabies? Maybe not. Maybe not. And at some point, I have to define what is the larger goal that I'm pursuing. Vaccination, yes. Mass vaccination and reaching appropriate coverage, optimal coverage, and finding a good vaccination model that can be scaled up. Yes, that's going to help me with eliminating rabies, and that's where I'm putting my efforts now. And when you talk about One Health, you definitely are talking about collaboration, and you are talking definitely about interdisciplinary projects, ideally transdisciplinary projects. And I hope you look for the definition of both and the difference between them. And this is our team. This is not up to date, so there are new people now joining the team, and some of the people in this picture left, especially because of COVID. Some of the people in the lab, they're working now in the Ministry of Health. In the Ministry of Health, they needed help, and they needed people trained in molecular techniques to do, you know, PCR and sequencing. So a couple of people from our lab, wet lab, left, and I'm glad that they were helping, you know, the Ministry of Health doing that important job. But yeah, it's because of them that, you know, we can do this kind of research. We almost always work just with primary data, so it takes a lot of effort in the field to collect those data. And of course, we collaborate with the local authorities. I think it's very important in global health to do that, and not just be, you know, the academic that takes all the information or makes assumption about the important research questions or the important interventions that are needed, and then they just do what they want. I think that you have to talk with them and see what their needs are, you know, and also make them part of the team. And thank you. And this is the actual view of This was the actual view of my room when I was living in Arequipa. So I hope that you have the opportunity to visit our site one day and see also this volcano outside of Arequipa. I hope I left time for questions.
Oh, thank you. That was great. Does anyone have any questions they would like to ask?
I have one question, just kind of about the role, you know, with the COVID pandemic and like the origin story and that relation to One Health. I was wondering if you think that that's going to help propel One Health kind of into the spotlight a little more, or where do you think that will go?
No, I think it will help. I think it will help. We already saw a huge increase in funding and increasing interest, you know, from from in every direction. We see more applicants, you know, to MPH programs, to PhD programs, more people. For the first time, I think that I received an email from a biostatistician interested in infectious diseases. Always the emails that I receive are from veterinarians and epidemiologists. And we usually look for biostatisticians to help us with the stats, but this time there was somebody actually looking for us, and so there is interest in them. The funders, yes. I don't know if you know about the new Institute of Zoonotic Diseases at Penn Vet, but that also just started, and it's, you know, because this is the right time to do that. And I think that now with all these discussions about human behavior, the origin of the virus, potential reservoirs, right? We're talking about deer, all that, people are seeing the connections everywhere. Now, before, sometimes for some audiences, we had to put very clear examples and multiple examples so people understand the interactions and the connections. Now, yeah, we don't need to do that. Everybody understands that everything is connected to everything.
Thank you.
I guess I had one question. When you were working with the kissing bug vector, and you kind of showed that data where you saw, I think you looked at the timeline of when they died. How did you detect parasite burden? Like, so when you injected the guinea pigs, how did you detect the parasite burden within the guinea pigs? I guess I was curious.
Yeah, so when we do these experimental infections, we know exactly the dose, right? We count the parasites in the inoculum, so you know how much you are putting in them. And then 2 weeks after, we take a blood sample from the animals and just also evaluate parasitemia. But we only wanted to know if the infection was successful.
Okay.
So 1 parasite or 1,000 parasites in a drop of blood, that was enough for us, because there is this chemiotaxis. So when you put a syringe, you get some blood with the parasites that were in the area. But when the kissing bug, you know, introduce their proboscis and they introduce their saliva, there is chemiotaxis, and some parasites look for that. So the kissing bug is going to obtain more parasites than a syringe. That's what I'm saying. And also in the kissing bugs, there's amplification, right? Because the parasite reproduces. So even a very small number of parasites is going to increase a lot of parasites in the feces later on.
Okay, so they multiply pretty quickly.
Yeah, actually, that's how this disease was diagnosed 30, 40 years ago in hospitals in Brazil and in Peru. They had kissing bugs. And if you came with all the signs of Chagas disease, all the symptoms, they would put 20 of these kissing bugs in your arm for 20, 30 minutes, you know, and when they were all engorged, they will save them for a few weeks. So the parasite reproduces, and then it was very easy to find in a microscope the parasite.
Does anyone else have any questions? All right.
Can I ask a question?
Yeah.
What is the background of the audience here, and what is are you in a program right now, like the MPH, dental, and vet?
Yeah, so I can start off. I'm in the public health program with a focus in global health. This is my first year. And my name is actually Brianna. I'm using someone else's link. So my name is actually Brianna. And I find this really fascinating because I'm actually interested in infectious diseases and want to kind of work in implementing like programs, prevention programs and things like that. So I think that this has been really easy to learn about.
My name is Caroline. I'm a second year MPH student on the global health track. And I took a class last semester in health security, and it really focused on the One Health aspects of health security. And so we learned a lot about that and about wet markets and kind of the need to amp up resources for One Health. So I also enjoyed your presentation to get more concrete examples from the vet side than like kind of the more public healthy side. So I appreciate you coming. Thank you.
Hi, thank you so much for your talk. I'm Anisha. I'm a second year MPH student, also on the global health track. I did my undergrad degree in economics, so I'm interested in sort of the intersection between global health, economics, and development, and I'm doing some work at Penn Center for Health Incentives and Behavioral Economics, and interested in behavioral economics in a global health setting.
Nice. That's very important. That's the thing. In global health, I think that there is a niche for everyone. You know, in global health, somebody told me once, I was explaining, you know, the different aspects of research, and not only my research, but my collaborators. And then this person asked, so everything is public health. This is what they said, and I'm like, yeah, pretty much. You know, you work in food, if you work in, I don't know, the environment, whatever. You know, there is always a connection with public health. So. Yeah, great.
Hi, I just want to introduce myself. Thank you so much for that talk. It was amazing. I'm Maria Cortese Herring. I'm actually the Assistant Director of Global Health Training for the Center for Global Health. And so, yeah, so I help these students be able to go do global health experiences. I was a global health track MPH at Penn myself. Your talk was fascinating. And if there are any other students on this call, please, if you could release your camera and let us know who you are. This was a great talk. Thank you very much.
Thank you. And it's fine if you cannot turn on the camera, but just want to introduce yourself.
I know. Oh.
Hi, I'm Guilhem. I'm a Spanish exchange student. I'm studying biotechnology in Barcelona. I'm here like doing my final degree project. I'm really interested in infectious diseases. So I really wanted to know your work and your studies, and I really liked your talk.
Thank you. Nice to meet you, Glenn.
Thank you.
Yeah, I didn't talk much about the technological aspect of my research, but I guess that more people now talk about that. But yeah, we do genomics and so yes, and so we have to, you know, play with bioinformatics and all that. But at the end, I think that global health is about people. I prefer to talk more about, you know, the connections with the communities and the connections with the health authorities. And at the end, I think that real change at the end is done at the people level. But yeah, we need tools and we need a lot of biotechnology and everything. I'm also the co-director of the certificate program in public health. And we have biomedical PhD students. And I like that when they are in this certificate program, they're exposed to other aspects of research and public health research, or just public health programs. because many of them are working about, you know, a specific protein that they're going to use for a vaccine, but then what are the implications for that, right? When you start thinking about that, I think that also, yeah, at least for me, I get more passionate about my research when I think of the communities that are going to be impacted by my research.
I guess I just had one final question. If you could just give us, I guess, advice on as we move on to like go out into the world and graduate from our respective programs, what advice would you give it to us starting off?
That's a hard one, hard question. One, create your own mentoring committee or your mentoring team, an informal mentoring team. But you need people who are going to advocate for you. You are going to you need people who are connectors. Everybody tells you you need to network. You need to network. For me, that was terrifying. You know, I think that I'm an extrovert, but I don't know how to network. You know, I can talk about a lot of stuff, but I don't know how to. And I saw people, you know, networking until I found a couple of people who were connectors and who believed in me, and they're the ones that every time that I told them I'm going to that institution, oh, let me send an email there to this person that I know, you know. So I have a couple of those in my informal mentoring team because I'm still junior faculty, and I know if you join a new institution, get somebody who knows the ropes and who also, you know, cares about you because you will need help. And that's going to make your life much easier. If you're going to academia, get somebody who understands funding. So I have all those people, and some of them are great just at one thing. I like them a lot, but sometimes I know one of them is very good at giving me advice about a specific thing. But if I ask about all stuff, I know that I don't want to follow their advice. So. But I mean, they care about me. They spend time with me, you know, and I think that I consider myself successful. Sometimes it's difficult to say that, but you know, I am like, yeah, I'm doing okay. You know, I never expected to be in this institution, and I never expected to get the funding that I'm getting and all that. So I have to say that, yeah, I'm successful, but it's because of all these people that are around me, you know. And I think that I was always trying to be close to smart people, caring people, you know, people who are smart but also humble, you know. It's difficult, but when you find those, just keep them on your side all the time. I think that's my best advice.
Thank you. Thank you so much for speaking with us today. We really appreciate it. Great presentation. And thank you everyone for attending. Have a good afternoon.
Thank you. Thank you so much for the invitation. Bye.
Bye.